So I decided instead of talking, I'll show a little bit of ICN3D live because it was frustrating to see only these 2D slides, right, without any 3D picture or 3D object. So I guess not everybody is back, but we can start. There are some people online. So we're going to have a panel discussion, and hopefully more people will interact about how should we go about creating an ecosystem starting from ICN3D, Molstar, and a few others to actually get functionality we need to study proteins. So here is an example of an AlphaFold structure that can be annotated on the fly by ICN3D. So for example, it will identify all the domains. Here we have 10 IG domains, and it will, give you kind of a rating with TM scores about what kind of domain it is exactly. Now, this can be improved, but it already works. And so interactively, you can build this. You can take any structure and annotate it as long as you have IG domain. Now, this can be done for other domains, but it needs to have the same kind of reference numberings that we've put in. So, in fact, this comes from, if you want to look at many examples of ICN3D in real research, you can go to bioRxiv, and we have a preprint that we just released here. So this preprint here will tell you what we do. And I'll present this kind of stuff at 3D SIG on Thursday.
You can click on help and then live gallery, for example.
No, I don't want live gallery because live gallery don't have the research behind.
Okay.
Well, there is research behind, but it's just, no, of course. So here in this paper, you have lots of examples of links that GIO was talking about and that actually Henry was talking about, because once he has a topic and a picture that he has annotated, he has a link. And clicking on the link will bring you something like what you see on the screen right now. Oh, you don't see this. How do I get?
So you can open another tab in that one.
Oh, so it's not the same browser?
It's a different screen right now. You have to, let me just.
Can you slide it? Oh, perfect. Yeah, sorry. I thought I was on the same screen. So, I mean, you go to that paper that's very new, and you will have a lot of examples there of all sorts of proteins and all sorts of analysis that we've done. So Barry's not here, but he's working a lot on IG domains, and let's say, for example, that you have, I'll take something simple here. So here is, for example, arrestin.
The link is in the caption.
So the arrestin, and here you would have, for example, open link in new tab. Did it work?
Yeah, okay. You have to just click on the tab next to it. Yeah.
Yeah. So here you see some features of iCn3D. So basically right now, when you're sharing a link, you're sharing the whole analysis. You're sharing the software, the analysis, and the visualization corresponding to the analysis. And here you have something that has been annotated already automatically as an IG domain. And you can read the paper about the details and then study how the IG come together, study how the two arresting IGs bind actually to GPCRs. And yourself can take that and do more analysis with it. So it's completely interactive in that way. Anyway, so for the panel, which is pretty light right now, we'd like to know what directions should we take to make life easy for biologists and structural biologists or biochemists to work with proteins. And so how much time do I have on the introduction? My time is up. So now it's Alyssa.
Huzzah!
You have to introduce her.
So Alyssa is actually helping us with all the hackathons. Come here.
I didn't know you were ready.
All the hackathons that we are running, that we've been running in the past. And she's been helping, she will help us put together this ecosystem. So please.
Right. Are my slides